So amyloid is a sticky protein that has this tendency to clump together, and it's produced from a large precursor protein, the amyloid precursor protein. We're trying to understand how it's produced, how it then clumps together, and how those clumps of protein interact with the surface of our nerve cells and then cause the problems that we see in people who have Alzheimer's disease. If we could understand how the amyloid is produced, how it's clumping together, how it's interacting with the nerve cells, then there's the potential there to disrupt those processes as potential treatments for the disease. And so a lot of what we do is just trying to understand the basic processes that are going on in the brain and whether we can then exploit any of those as a potential therapeutic target for the future development of drugs. So one of the other projects we've got is around understanding how the clumps of amyloid protein stick and interact with nerve cell membrane. And it's a particular project funded by Alzheimer's Research UK to look at the mechanisms underlying that and to look at whether we can disrupt those mechanisms in any way. And this is a fairly complex project. It involves a range of different techniques, including very high resolution microscopy, to actually look at the surface of the nerve cell and at how these clumps of amyloid are interacting at the surface, and to say whether we can potentially disrupt that in some way as a potential therapeutic option going forward.